delighted to have Adeline Vanderver here, who has really led the charge in the GLIA clinical trial network, and we'll be talking about progress there. Thank you so much.
Thank you very much. And to speak to Florian's last point, I think one of the issues, specifically with leukodystrophies on the African continent, is that most places don't even have MRI machines. And most of our disorders are ascertained by that type of imaging, and so you can't even get at families, and people are then misdiagnosed with cerebral malaria or whatever other is the diagnosis of choice because of lack of access to diagnostics. So it's a really complicated issue. So first of all, thank you so much for inviting me. It's so nice to be here, and I'm sorry that I'm jumping in and out a little bit today. And I also want to just say how impressive it is, the size of this room. I feel like every year this meeting has more and more wonderful attendees, and it's a pleasure to be part of them. So I'm going to speak a little bit about a joint effort between myself and Florian and Ali Fatimi, as well as others in this room, to try to create a network to improve clinical trial readiness. And it was really, to step back for a second, driven by our patient advocate partners, because they're the ones who in 2013 asked us to start thinking about forming a collaborative network that would be eligible for funding with the RDCRN, and I want to give them credit as well. But I think that this presentation is meant to give you a little bit of an idea of what the structure is of this program, and also what the participants are, and just so that everybody's sort of familiar and at the same level, understanding what the project entails. So this is a project that's very much driven by NIH rules. It's part of something called the Rare Disease Clinical Research Network. There are about 24 different research networks that cover a span of different rare diseases. People are eligible for resubmission, so many of these networks go on for many years, up to 15 years total, and really gives us a chance to build a hopefully sustainable network in which we can collaborate. And the goal of the resources that are given by the NIH is more to drive infrastructure support than specific projects. The specific projects are almost a way to develop that network, which then hopefully can be funded additionally in other ways. So it includes an administrative unit, right, which includes myself and Florian and Ali, as well as Josh Bonkowski, and then many of you know also Omar Sherbini, who is the program manager for this and who helps us with our organizational skills. We're going to be adding to that administrative unit with additional support persons to improve our communication. So we'll have a specific person who'll be working with advocacy and communications, as well as adding additional sort of technical level support over time. We also have multiple sort of groups within that network. The first really important one is a data integration core, which is a partnership between some of the data management team that Florian has used so effectively as part of ALD Connect through Neurobank and through Alex, who I saw briefly in the hallway earlier, although I don't know where he is in the room now, as well as the teams at CHOP, who have expertise in a specific approach, which is to basically query the medical record to try to get access to big data. And you saw already with Josh Bonkowski's one approach at looking at big data through these sort of national registries, but there's an increasing number of ways of doing that, and CHOP has had expertise and already participates with a number of the institutions that are on the RDCRN application to collect that type of data, particularly at this point in the oncology world. We also are working with a group of a large representative group of the advocacy partners. We tried to have a broad outreach. The NIH has told me with some consternation that we have the greatest number of different advocacy partners within our RDCRN, and so I think I really look forward to that interaction and to the fruits of sort of having all those multiple different perspectives across the different disorders and working with those partners. We want to make sure that, I think to quote one of our advocacy members, we want to make sure that the RDCRN GLIA-CTN is a we that includes the advocacy partners, and that they are more, that there's a presence that is felt really throughout the process. And to that point, we're being very thoughtful about including RDCRN partners that for all the committees that will include advocacy members. The start of that collaboration will occur at the meeting that's going to occur a little bit later in November. There's an RDCRN CPAG global sort of meeting, and we're going to meet with a couple of the groups that are able to make that meeting to sort of kickstart that process. And then finally, we want to recognize that we're at a very pivotal point within research in the leukodystrophies that includes and needs participation with industry partners. They have resources and access to protocols and approaches that we don't have within academia or within advocacy members groups to be able to really drive therapeutic development. And so we're open to those collaborations that will be thoughtful and will be proceeding in ways that make sure that we keep ownership of data and processes within the academic and CPAC side, but really want to embrace also those partnerships to make sure they're fruitful and we get them the data they need to advance the causes you all have in mind, which is to develop therapies. I also wanted to speak for a moment about the current participating institutions in the RDCRN, and I wanted to emphasize that these are our current partners for this effort. The choice of those was driven somewhat by geography. You can see that they're distributed to some degree across the United States, although there's always a little bit of an overrepresentation of the Eastern Seaboard. But I also wanted to comment on the fact that we know that there are many of you with whom we have close collaborations who are partners at other sites, and that the choice of the inclusion of those sites, including international partners, was driven by a pragmatic sense of what we thought the reviewers would be likely to see as feasible. So the sites that were chosen have features including access to things like CTSA grants and other large NIH grants that would make it a convincing story that we would be rapidly sort of be shovel ready and able to leverage this as a functional network. And we don't want people to think that that means we don't want inclusion of other international and US-based partnerships, and we're already reaching out to some of you to extend those efforts beyond the specific RDCRN funding mechanism. And then not least, we have significant investment and leadership from our advocacy groups. And again, I want to give credit to the advocacy groups that were part of our first 2013 meeting, including the PMD Foundation, Hunter's Hope, and the MLD Foundation, and who really have helped drive this process from its inception, and with whom we're honored to continue to partner as we develop some of these processes. I want to go back also to that original 2013 mission statement, which is a little bit dense in length of text, but emphasize the way that we really want to use this as a resource to pave the way for transformative therapies. And so that the term clinical trial network really emphasizes that we want to be clinical trial ready. We want to create tools that make it easier and decrease the bar that people need to leap over to do clinical trials and develop therapies. So the project then leverages that infrastructure, right, which includes those multiple sites, to do several types of exemplary projects. And the first is mandated by the NIH and is a large natural history study. And I'm going to go into it a little bit in detail, but they want to have demonstration that you can do natural history and you can demonstrate that you can collect longitudinal data. The second is a project on AMN, which will be the closest and nearest to everybody's hearts here, and is led by Florian and Ali. The third is a project in Alexander disease, for which the need was emphasized by Albie Messing's recent publication showing that antisense might be an effective therapy in this disorder, and with imminent clinical trials, the need for clinical trial readiness. And similarly, the fourth is for Aicardi-Gouttières syndrome, which with several planned and ongoing clinical trials in that disease sort of emphasize the need for a better understanding of the longitudinal basis of that disease and clinical outcome assessments. So project one, we have divided into three aims. The first aim is what people would think of as a standard natural history study, which is you bring families and patients into a clinical setting, you measure functional scales like how fast people are able to walk or how well people are able to use their hands that are well established, acceptable to sort of researchers as far as tools, and basically assess how easy those tools are to apply in specific diseases. And here we picked detect a series of diseases that, again, just like I mentioned, the centers are not representative of our hope and breadth of diseases overall that we will impact with this, but more just because they're exemplifying the age span and the type of disability seen across the leukodystrophies overall. So it's our hope that as we demonstrate these tools in HES, Alexander disease, MLD, Plessis-Merzbach disease, and AMN, that we'll be able to then build on those lessons learned to then expand those to other disorders with similar levels of disability. And those patients will come every 12 months and be followed at the home centers, i.e. closer hopefully to where they live, for those outcome assessments. The second aim is one that brings those assessments closer to home. So we're planning remote assessments in the patient's home through use of video approaches and through use of motor mobility assessments with automated systems, and also through use of parent-reported surveys to sort of compare what we can do infrequently in the hospital care setting to a more frequent setting in the patient's home. And then the one example of this is the actigraphy, right? And I'm sorry, I'm skipping through because I'm being thoughtful about time this morning. The actigraphy is a wearable device, for example, that can give measurable information on sleep and on level of mobility and activity in patients without necessarily the patient having to see a physician or a physical therapist. And the important thing is that we hope that over time this will diminish the need for patients to come into a medical setting for follow-up of a clinical trial, for example. And if we can demonstrate good correlation between that first part that I was talking about, which is the sort of the standard, gold standard approaches, and these type of in-home, patient-reported and wearable devices assessments, it'll lower the complexity and the cost and also the burden for patients of clinical trials. And then the third aim is an aim that is based on collecting data from medical records to sort of catch up with our need for natural history. You know, we're in such a rich period right now developing clinical trials that we have more potential therapeutic platforms that could be applied to these group of disorders than we ever could have time to do prospective natural histories. And sort of my worst nightmare is having a therapy ready now and having somebody say, oh, but now we need to spend 5 more years understanding how the disease progresses before we can actually apply that therapy and test it. And this is an attempt of a demonstration project to show that we can go back in time using the medical record to actually collect that longitudinal data to understand how the disease progresses with enough detail that that can be used to understand how to plan clinical trials, and in some cases even use it as non-concurrent, what's called non-concurrent historical controls. And that requires a complex data management system, which will include both manual extraction and automated extraction from medical records by a team of a mobile team that will go out to the different sites as well as interact with the information technology groups to automate, in an automated fashion, take data from the medical records of patients at specific sites with consent. And just as an example, this is a project that we've been doing now for about a year in MLD where we've done that effort. And you can see, I don't know if this, oh yes, it works. You can see, for example, that level 5 is a child with MLD who's lost the ability for basically all motor control except for neck control. So it's a child who goes from a normal level to a level that really has significant functional disability. And you can see that by age of presentation, if you present before 2.5 years, you're going to very rapidly attain that stage. Versus if you present later, you might have a much more slow progression. And that specific approach has allowed groups to talk to the FDA and say, you know, we don't necessarily need to have a control group in our trials because we already know how badly the children are going to do. And if we manage to shift that, that's a win, right? And so it can have very meaningful impact. So project 2, right, shifting gears now, is an AMN-focused project. And it was built off of Florian and Ali's need to understand how to measure some of the disease features of the spinal cord, some of the myelopathy. And so specifically, they have developed tools to actually look at mobility and to look at to what degree people are also having challenges with balance, right? And so by using both assessments at Kennedy Krieger and at MGH, and you need to interrupt me if I'm butchering the actual application of this, but also comparing it with then wearable technologies done again in the patient home, the hope is to demonstrate that there's good correlation between some of these measures of ataxia and measures of movement, both in a hospital setting, but also good correlation with measures at home, again to pave and ease the way for clinical trials in this group of in this group of patients. And in this particular trial, there's a confirmed diagnosis of AMN, right? A basic ability to retain walking, right? And some degree of functional disability without medical features that would make it very burdensome for the patient to participate in the activity. All right, so project 3. This is a project on Alexander disease and is focused again on the need to understand biomarkers specifically in Alexander disease. So in AIM-1, sorry, I went too fast. In AIM-1, we're going to work on measuring a specific biomarker in both blood and CSF, that's GFAP, which is the protein that's abnormal in Alexander disease, and will allow us to understand how easy it is to measure at multiple sites and across time that GFAP in both blood and CSF in patients to prepare for clinical trials. And then in AIM-2, understand to what degree that level of the GFAP is actually correlated with clinical features, because it doesn't matter what a protein measurement is if it doesn't match what's going on with the patient and their health. And so looking at how we can correlate that protein with Alexander disease subtypes at presentation, as well as going back to that bigger project from the first project, understanding how is it predictive of how people do over time. And then the final project is based on AGS outcome measures. And in that project, we're going to try to understand acardic Gucci syndrome is a very heterogeneous leukodystrophy, with some patients presenting as tiny little babies and other people presenting as adults, very much like you see in ALD and AMN. But we don't yet have the understanding that you have an ALD and AMN about what boundaries there are between those different presentation groups, and so we need to do that work, as well as understand if we can develop a disease-specific rating scale. Again, those are things that are present already in ALD that we don't have at all in AGS and that we're working to develop. And then finally, understand again the proportionality between a specific marker of disease, which is an interferon score, and how people do over time. I'm going to speak briefly about administration, so that if at some point we reach out to you for help with some of these procedures, you understand what the structure is. As I mentioned briefly, there's an administrative unit, right, and a program manager administrator, and those groups make up part of the executive committee, but not all of the executive committee, because the steering committee also will include representatives from the advocacy groups as elected by the advocacy committee, and also representatives from the different other subcommittees, as well as some of the site PIs from the participants of the different projects we just discussed. And importantly, the advocacy committee will need to help us with leadership on both their own committee and sort of defining what their role should be as part of the CRCN, but also defining who and in what capacity they want to be represented on the other committees. The other committees, the committees will include the data governance, pilot and education committees, and as well as the external advisory committee. So Florian very nicely sort of tried to define for us in the past weeks what does success mean for the GLIA-CTN, and I think this slide somehow got out of order, Dean. That's probably, I'm sure it's my fault. Okay, I'm going to come back to this slide in a second. The GLIA administration also includes that data integration core I spoke about before, and data governance. And I wanted to speak briefly about the data integration core and sort of speak to the fact that these are this will essentially create a data warehouse that will be represented also by data that will be housed at MGH. That's part of the work done in ALD, but also across some of these other diseases. And we hope that this will serve as a resource for the community. So this is not something that I see as being something that only the members of the GLIA-CTN can participate in, but also hopefully serve stakeholders outside of that. And we would welcome the opportunity to understand what projects you think you could potentially leverage this type of infrastructure for, because we're building the infrastructure to enable other projects. There'll also be a pilot study committee and a career development committee. We're very committed, both as a mandate from the NIH, but also as a personal objective. I want to be able to retire someday, which means I really, really want young people to be interested in this disease and to be able to be successful in research in this disease. And so we'll be very focused on the pilot studies in career development. Specifically, the first pilots will be based on the work that we did at the GLIA meeting in May. We've already outlined, based on everybody's feedback, a number of key things that the disease community feels needs to be advanced, and we'll be providing more information about that soon based on the surveys and the other work we've done over the summer. And then there is this external advisory committee, which will hopefully allow us to leverage the experience and the support of other groups that have successfully done this type of effort before, and we will listen carefully to their advice. And then finally, this Industry Advisory Council will hopefully permit and give a platform to our industry partners to provide information and guidance to what projects they think are the most important. So that the not last slide, because the last slide I'm going to go back a couple, this is sort of the overall organizational structures and explains the fact that we are answerable to a certain amount of sort of NIH oversight in this project, that we hopefully will be able to integrate the information and the desires of all our sites, but also very much the patient advocacy and industry advisory groups. So I'm going to go back. Sorry that the slides were out of order. So what is success, right, for this group in the end of the day, right, is to really establish a clinical trial ready network, right, through collaboration. And we want to make sure that by doing so, we define appropriate clinical outcome measures, define meaningful biomarkers, define the rate of change, standardize these things across multiple sites, and build an open and engaged community. So I hope that you'll be willing to partner with us, and we look forward to working with you.
Thanks so much, Adeline. There's time for questions before we go into our panel. Just to mention that as part of Project 2 on adults with ALD, this will include both men and women, and I think that's really important. And I think it's going to serve, lead to, I think, the next phase of studies and trials here. So I think very nice opportunity to breathe a little bit outside of the ALD community as well, and have feedback and create some collaborations across different diseases.
Yeah, we very much hope that the lessons learned in AMN will be helpful also for other leukodystrophies with myelopathy and other adult presentations of leukodystrophies.
Good. Thank you, Adeline. Okay.